Hello, welcome to Meiosis World. Today you will go on a trip through meiosis. Here we go. The first step of meiosis, just like mitosis, is interphase. This is when the majority of cell growth is. The cell synthesizes new proteins and organelles, and DNA is replicated. And now, here are the steps unique to meiosis. The first step is prophase 1, where homologous pairs of chromosomes are formed, creating a tetrad. The crossing over occurs between this tetrad, where homologous chromosomes exchange portions of chromatids producing new combinations of genes. The tetrad is made up of one maternal and one paternal chromosome. These chromosomes exchange chromatids, which is important for genetic variation because the offspring will look like both the mom and the dad. Coming up around this bend, if you look up to your right, you will see metaphase 1, where the tetrads line up along the middle of the cell while remaining in pairs and the spindle fibers attached to the centromeres. Now coming up is anaphase 1, where the homologous chromosomes are pulled to opposite sides of the cell by the spindle fibers. That was telophase and cytokinesis 1. At the end, each set of homologous chromosomes have been separated into two daughter cells. The nuclear membrane and the nucleolus reform, the cytoplasm divides and cell membrane is pinched, which creates two haploid daughter cells. That concludes meiosis 1. For meiosis 2, some cells skip interphase 2, and there is no S phase because DNA does not need to be replicated. Coming up is prophase 2. The nuclear membrane and nucleolus break down. Spindle fibers form and the centrioles move to opposite sides of the cell. This is just like prophase 2 in mitosis. Just ahead is metaphase 2. Chromosomes line up along the middle of the cell and the spindle fibers attach to the centromeres. Next is anaphase 2. The sister chromatids split and are pulled to opposite sides by the spindles. The last phase in meiosis 2 is telophase and cytokinesis 2. This is when the chromosomes uncoil, the nuclear membrane and nucleolus reform, and the spindle fibers break down. This results in 4 haploid, genetically different daughter cells. And there they are. The cells in your body that go through meiosis are not your normal somatic body cells. They're your reproductive cells. For the men, sperm. For the females, eggs. For the men, meiosis is important because it creates 4 haploid sperm cells from one spermatogonium. The reason more sperm is important for men is because not all sperm will make it to the egg. The more there are, the better the chance of the egg being fertilized. For females, The egg divides unevenly so that the bigger cell has enough cellular content to sustain life upon fertilization. The smaller cells are discarded from the body. Although the bigger cell is bigger, it still is haploid and has the same number of chromosomes. When an egg and a sperm meet and are fertilized, it becomes a zygote. This zygote will then undergo cell division through mitosis to develop into an embryo, which will grow into a fetus, which will grow into a human. Thank you for coming. We hope you enjoyed your trip through meiosis.